Hello, class. Welcome to the next unit, Unit 3. In the last unit, we talked about chemical reactions, enzymes, ATP, cellular respiration, and photosynthesis, ways the cells get energy and make food in case of plants. Now, we are going to talk about DNA and protein synthesis for this week. Later, we're going to talk about what's called cell division and more topics. So today or this week, we're going to talk about DNA, its structure. We're going to talk about genes and how proteins are made. So what is DNA first? DNA is a macromolecule, one of the big molecules in the cell. And we have four types. We have carbohydrates, proteins, lipids, and nucleic acids. Each macromolecule is made up of what's called a monomer. Monomers are the building blocks of the Legos that are put together to make these giant molecules. So DNA is one of the types. It is a nucleic acid. We also have RNA that's also a nucleic acid, which we'll get to in a second. It's very similar to DNA. The monomer for nucleic acids is nucleotides. And this right here is a nucleotide. And we're going to break that down on the next slide. The big function of DNA is DNA stores the information that the cell needs to produce protein. It is the cookbook for the cell. It has all the instructions the cell needs to make all the proteins it has. So when a cell needs a certain protein, it's going to look at DNA, take these letters, and find the instructions to make that specific protein it needs. And before we get into that process, though, let's break down DNA, starting with its monomer, nucleotides. So again, this is a nucleotide. And we see it has three parts: phosphate group, sugar, and nitrogenous base. DNA stands for deoxyribose nucleic acid. Deoxyribose is the sugar molecule that it has. Remember, a sugar is a carbohydrate. So there is a sugar molecule or carbohydrate within the nucleotide itself. And the specific one is deoxyribose in DNA. We also have this yellow phosphate group on the left, and then we have the nitrogenous base on the right. This nitrogenous base is the part of the nucleotide that can change. We're going to go over the types of it in a second. But in general, these two parts, the phosphate group and the sugar, do not change. But the nitrogenous base can change. Now, with our nitrogenous bases, we have five types. And in general, they come in two categories: pyrimidines and purines. Pyrimidines have a one-ring structure, purines have two-ring structures. Pyrimidines are cytosine, uracil, and thymine. And you can see each of these has a little letter, C, U, or T. So pyrimidines are cut, while purines are the two-ring structures. We have adenine and guanine, or AG. So these are the different types of nitrogenous bases that exist within DNA and RNA. Keep in mind, though, that CAG are DNA and RNA, but uracil is only in RNA. Thymine, or T, is only in DNA. We're going to see why that's important later on today. But notice we do have five types of nitrogenous bases, but one of these is present in DNA, one is only in RNA. So now, nucleotides join together into strands of DNA. So again, nucleotides are the Lego pieces, the building blocks of DNA, of nucleic acids. So we can put them together to make a big molecule, the DNA. The nucleotide sequence of DNA is the order of the nitrogenous bases in the strand. So when we talk about nucleotide sequence, we're talking about the letters, the nitrogenous bases in their order. So we can see here, this blue part is what's called the backbone of DNA. And we have the phosphate and the sugar that make up the backbone, while the nitrogenous base is in the center. So the nucleotide sequence is just the order of the nitrogenous bases as we go down. And in this example, we have C, G, T, C, G, A, and so forth. So that sequence is what the cell uses as the instructions on how to make a protein. That is the language of DNA, these nitrogenous bases. DNA is also what's called a double helix. One molecule of DNA is made of two strands of nucleotides. The two strands are wound together into a helical shape. So you can see here, we have one strand that goes this way and one strand that goes this way. They are joined together, and these are hydrogen bonds that are joining them together by their nitrogenous bases. And when they're joined together, they're twisted into this shape, this double helix. Double because it has two strands, a helix because it's twisted. So DNA put together makes this double helix formation. DNA strands are held together by base pairing. Like I mentioned earlier, hydrogen bonds between nitrogenous bases connect the two DNA strands. But this is the key thing that I want you to know. I'm gonna get my pen out for this. Know these base pairs. So whenever we talk about base pairs in DNA, adenine binds with thymine, guanine binds with cytosine. A to T, C to G. A to G, A to T, C to G. They will never mix. You will never have a G to a T, or G to an A, or an A to a C, or A to C, G. You will never have that. It's always G and C together, T and A together. And that's important because when we have this double helix formation with the two strands of DNA, C is always with G, T is always with A. So if one strand you have a C, you might automatically know, oh, on the opposite strand, on what's called a complementary strand, we have a G. If we have an A on one strand, we know, oh, on the complementary strand, the other strand, we have a T, because A always goes with T. So the base pairs are always going to be like that. A to T, T to G, or T to A, C to G. Make sure you understand that part because we're going to use this later on. DNA strands, again, are complementary, kind of like how I talked about. Hydrogen bonds can only form between complementary base pairings, so A to T, C to G. Nucleotide sequences on one strand determines the sequence of the other strand. And that's exactly what I talked about. How if you have a C on one side, you know you have a G on the complementary strand. If you have a T on one side, you know you have an A on the complementary strand. They're always the same base pairs. A to T, C to G. Now, each strand of DNA is different. We have what's called a 5' area site and a 3'. 5' is where the phosphate group is, and 3' is where this OH group is at the bottom. So phosphate is 5, the sugar is 3. So when we look at this, we're going down, we notice that we hit phosphate first. So this means this side we're going from 5 to 3. Phosphate, then sugar, phosphate, then sugar. 5 to 3, 5 to 3. The other side though is actually the flip. The strand is reversed. So if we go down, we have 3 to 5, 3 to 5, sugar to phosphate, sugar to phosphate. Because remember the sugar, we have the 3 at this carbon, and the phosphate we designate with the 5. So the strands are anti-parallel, oriented in different directions, with one upside down with respect to the other. So that's going to be important. When we look at DNA, one side will go to 5 to 3, the other is 3 to 5. So describe the components of DNA and its three-dimensional structure. Pause this video, see if you can describe DNA and its structure, including its shape, the nucleotides, the base pairings, and the direction of the strands. Okay, let's continue. So, protein production starts with DNA. We talked about DNA, we talked about how it's nucleotides put together with the bases being aligned. A gene is a small region of a chromosome. A chromosome is a tightly packed DNA. Tightly packed DNA. So you can see here, we have our DNA here. It's wound together through different proteins. or even tighter together into what's called a chromosome, which is that typical X shape you see with DNA. So a gene is a small region of the chromosome. It's a sequence of DNA, and each gene encodes for a specific protein. So the gene is the actual instructions on how to make a protein. I mentioned earlier, think of DNA as a cookbook. The cookbook holds all the instructions how to make the proteins in the cell. The gene is the one set of instructions for a specific protein. So you take that one gene, you can read it from the DNA to make a new protein. Now, we do need to talk about RNA though. RNA is another nucleic acid and it's slightly different from DNA. We can see in terms of the sugar, we talk about the nucleotides and DNA have deoxyribose. RNA, RNA stands for ribonucleic acid. It's ribose because we use ribose as a sugar. So in a deoxyribose, we use ribose. In terms of the bases, instead of thymine, we use uracil. So uracil is replacing thymine in RNA. U replaces T in RNA. That means A binds with U in RNA. A binds with U in RNA. The form or the structure. We talk about DNA as a double-stranded helix. But RNA is only a single strand. It's only one strand and that's it. If you don't have a complementary strand, it's just one strand. Function. So for DNA, I want to summarize this function. DNA is the set of instructions stored in the nucleus on how to make a protein. It is the cookbook of the cell. RNA is the copy of the instructions on how to make a protein. So it's not the full cookbook. It's if you were to take a cookbook, look at one set of instructions and make a copy of those instructions, that copy is the RNA. So RNA is the instructions on how to make a protein, but it's copied from DNA. We have our full cookbook here with all the instructions, and we have the copy of a single recipe. A single recipe for one protein is the RNA. So remember these differences with DNA and RNA. RNA has a ribose as a sugar. It uses uracil instead of thymine, U instead of T. It's single-stranded, and it's a single instruction for making protein. Now, making proteins is like making a brownie. So with this, this is a key thing I want you to know. We're going to start getting into how we go from DNA to protein. So in this unit, I do want you to know each of these steps, what's happening, and some of the key players. And we're going to use the cookbook example still. So with the cookbook example, we have a cookbook. This is the chromosome with the multiple genes, the multiple instructions. We talked about how RNA is the single set of instructions, the copy. So what we're doing in this example, we go through what's called transcription. We take the instructions, we make a copy of them, and we get a single set of instructions. So in this example, we're taking the cookbook, we're making the copy of the recipe for brownies. That copy is the RNA. The copy of the instructions is the RNA. The RNA then goes to the next part, which is translation, where we take our recipe, we take our building blocks for proteins, which is amino acids, put them together to make the protein. And again, if we use a cookbook as an example, we have a recipe, we use that recipe to get all the necessary ingredients, put them all together to make what we want, the brownies in this case. So transcription is going from DNA to RNA. Translation is going from RNA to protein, using that instruction to build the protein. Again, transcription is RNA synthesis, translation is protein synthesis, and they work hand in hand. So transcription is RNA synthesis, excuse me. Transcription uses DNA as a template to build RNA. The nucleotide sequence in DNA determines the nucleotide sequence in RNA that is transcribed. Sequence is complementary to DNA sequence. So just like how we talked about DNA, since it's double-stranded, it has base pairs, A to T, C to G. we're using that base pair idea to go from DNA to RNA. And to do this, we have a couple key players. Some of these we're not going to see right away, though, in transcription. Well, right now we're talking about translation. So translation takes place in the ribosomes. So transcription is in the nucleus, translation is in the cytoplasm here. And we use what's called ribosomes, which we talked about briefly in Unit 1. Ribosomes are the organelle that makes proteins. There are 3 types of RNA, though. We have the messenger RNA, which the messenger RNA is the copy of the instructions. It brings the information to the ribosome. Ribosomal RNA makes up the ribosome. So whenever you see rRNA, think this is just the ribosome. It's the factory that makes the protein. And then we have transfer RNA. T for transfer because it's bringing the amino acids to the ribosome. It's bringing the building blocks. It's bringing the ingredients to the RNA, to the ribosome. So you have mRNA is the message, the instructions. rRNA is the ribosome, the factory that makes the protein. And tRNA is the transfer RNA, which brings the ingredients to the ribosome. So what is the main function of DNA? Pause this video and think about it, and we'll come back. Okay, so DNA encodes for proteins. It is the cookbook for the cell. It is a cookbook on how to make proteins for the cell. What is the relationship between a gene and a protein? Pause, think about your answer, and we'll come back. So a gene is the single set of instructions for a protein. It's the DNA that is a single set of instructions for a protein. It tells you how to make the protein. Now we're going to go over more in depth transcription and translation, starting with transcription, since that's the first step of this process. So transcription uses DNA to create RNA. So let's first look at how the cell produces RNA, RNA copy of a gene. And again, the transcription starts in the nucleus. So we're never in the nucleus because that's where DNA is found. RNA nucleotides pair with DNA nucleotides. To make RNA, base pairing takes place between RNA and DNA. DNA pairs with RNA. And just like we have A to T, C to G, we have the same idea. But instead of A to T, we have A to U because T is being replaced with U. So when we go from DNA to RNA, we have A to U, C to G, G to C, and T to A. Same idea as last time, but instead of T, we have U. A to U, or T to A, or C to G, or the reverse, C to G, or G to C, excuse me. Now, to break down transcription again. Transcription is a process of breeding a gene and DNA to produce the mRNA, or the messenger RNA. The messenger RNA is just a copy of the instructions. It happens in the nucleus, since that's where the DNA is found. And the molecules involved are 3. We have the DNA, of course, because that's the instructions. We have the mRNA, of course, because the copy of the gene transcribed into RNA. But we have what's called RNA polymerase. RNA polymerase is a big enzyme that reads the DNA to produce the correct mRNA. So this is the actual enzyme that takes DNA to make mRNA. This is how it works. So we have our DNA here. This big structure is the ribosome. Sorry, not ribosome. This is the RNA polymerase, and it's opening up DNA. It's reading DNA, it's transcribing it into an mRNA, and that mRNA, that message, is then sent off to do its thing. Remember, mRNA is a single-stranded molecule. So it's just one strand of nucleotides. So again, DNA, we have RNA polymerase, opens it up, reads the DNA, makes the appropriate mRNA, and sends the mRNA off afterwards using the base pairing T to A, A to U, C to G, G to C. Now, we are gonna do an example. For this example, you will be asked to do this on the test. For the lab this week or the activity this week, you're gonna have time to practice this, but you are gonna have to take a DNA strand and make RNA with it. And in the next part, we're gonna take that strand and make a protein with it. If the DNA template strand has the following sequence, what would the nucleotide sequence and the complementary RNA molecule produce in transcription? So let's look at this. We have A, G, T, C, T. So I'm going to copy that to a Word document real quick, just to make our life a little easier. We have A, G, T, C, T, T. So if we go letter by letter, remember, A to U, C to G, C to G, G to C, T to A. So A pairs with U, because we don't have thymine anymore. We don't have T anymore. G and C always go together, so G to C. T still goes with A, because we have A in RNA. C goes with G. T, T, that goes with A, A. So our answer should be U, C, A, G, A, A. U, C, A, G, A, A. We're doing one letter at a time. A goes with U, T with A, and G, C always go together. Now let's talk about translation. So translation is building a protein, taking the mRNA and building the actual protein. All cells have the same genetic code. All cells have the same genetic code. When we look at um, the mRNA, we break it down into three parts each, three nucleotides. These three nucleotides together form what's called a codon. A codon is a three nucleotide sequence that encodes for one amino acid. So here we have A, and G, they code for lysine. U, C, A, they code for serine. G, U, C, they code for valine. So every three nucleotides codes for one specific protein, one specific amino acid, I should say. And that's how we go from mRNA to a protein. And with that, when we break it down, this is what's called a codon table or genetic code, where the way we use it, on the left side, we have the first letter. So in this example here, A is the first letter. So we find A. At the top, we have the second letter. So we go A to the third column, A here. And then the last letter is G. So we find AAG. We go down, AAG is the bottom. So we get lysine. And that's how you go from the genetic code to the amino acid using this table. First letter is on the left, second letter is on the top, third letter is down the row, or down the column, excuse me. We do the next one, U, C, A. We start with U, right here. We go across the C, and then we do A, so UCA, we get serine. So this is how we use that table to determine the order of the amino acids. That's important because it's part of what's called tRNA. tRNA translates genetics code. tRNA is transfer RNA. It's a molecule that brings the amino acids to the ribosome. tRNA are adapters that recognize the genetic code. Anyway, with codons, they are these three nucleotides in the mRNA. So every three is one codon. tRNA has what's called an anticodon. This is the complementary strand of the codon. So here we have AAG, we'd have UUC as the codon, the anticodon, excuse me. So this is going to have an anticodon to know what three bases in the mRNA it needs to bind to, and it's going to have a spot over here for amino acid to bind. But remember, tRNA transfers the amino acid to the correct location in the instructions. So tRNA binds to the mRNA codon at the anticodon. and brings the corresponding amino acid. So again, this anticodon binds to the codon on the mRNA, the 3 base pairs on the mRNA, to bring the correct amino acid. So each step in translation happens at the ribosome. Ribosomes help the 3 types of RNA interact with each other to build a protein. The large subunit binds to the tRNA. So we have the large subunit here. The small subunit binds to mRNA. So when we look at this, the mRNA is going to bind to the small subunit. The tRNA is going to bind to this large subunit. The small one will prep the mRNA to be read, and the large one grabs the tRNAs to put all the amino acids together. And this is, again, just an overview of everything. Translation is the process of using mRNA to produce a new protein. It happens in the cytosol or the fluid outside the nucleus. We have 3 molecules or a couple molecules involved. mRNA. mRNA is the message, the instructions on how to make the protein. Ribosomes. Ribosome is the factory that makes the protein. It's the molecule that reads mRNA and starts the assembly of a new protein. tRNA or transfer RNA is the molecule that transports amino acids to be used to make a new protein. And lastly, we do have our amino acids that are the building blocks for protein. We have 20 different types. I'm not going to have you memorize the 20 different types, but now we have 20 types of amino acids. This is kind of how it looks together. So we have the mRNA here. We have the yellow is the ribosome. This purple is the tRNA. And we can see here, we have our 3-letter code. It's the codon. The tRNA has the anticodon, which is the complementary basis. And then on the other side, it has the amino acid. tRNA will enter, read the code. If it's the correct, it stays there. It keeps going until the next transfer RNA comes in, reads the code, and then if it's correct, the 2 amino acids will put together to start forming the whole protein. And this process keeps going, reading each 3 letters to have the next amino acid, put it together until we get this long strand of amino acids. So we're using all these transfer RNAs, these tRNAs, to come in, read the codon, and then attach their amino acid so we can make this long strand of protein. Oops, too far. So translation is efficient though. Multiple ribosomes attach to mRNA molecules simultaneously, so the cell can make many molecules of protein at once. We don't just have one ribosome reading each mRNA. We have multiple ribosomes that are working together to read the mRNA. So we have multiple of these ribosomes that attach to the same mRNA, and they're all building multiple proteins at the same time. That allows the cell to be more efficient. Instead of just waiting on one ribosome to make one protein, we can have multiple ribosomes reading the same mRNA strand to make multiple sets of that same protein. I like this video. It kind of shows you a little bit of how it works. Transcription and translation. So I want to start again with this. At the beginning, we have DNA. RNA polymerase comes in, opens it, makes mRNA the messenger in transcription. That goes to step 2, translation, where we have ribosome grabbing the mRNA and tRNA coming in, reading the mRNA to put the amino acids together in the right order. If you want to see another example, this YouTube channel, Amoeba Sisters, has a great video of transcription and translation. So I highly encourage y'all use them as a good source to help you look at this process in another way. So look at the image below. Which ribosome has been on the mRNA for the longest? Look at the polypeptide chain to see which one has been there the longest. So with this, if you look at the RNA, the amino acid chain, we can see these are nice and small. They're getting longer and longer as we go by. We get these big, longer strands. So the ones on the far right are the ribosomes that have been on the RNA strand the longest. So now, if the DNA strand has the following sequence, what will be the amino acid sequence of the corresponding protein? Just like I said earlier, I do want you to make sure you know how to go from DNA all the way to protein. So discard this. I want to take this strand. We're going to go over here and build it out. So whenever you go from DNA to RNA to protein, what I first want you to do is take every 3 letters and put a space. Because remember, every 3 letters is a codon in mRNA. So we're going to start that with the DNA. So we have our DNA strand here, and I'll label that here, DNA. What we're going to do is we're going to make mRNA first. Okay. So remember, C and G always go together, but A goes to U, T goes to A for mRNA. So we start with A. So we go letter by letter from DNA, not Dan, ignore that. Autocorrect. So we start with A, and our mRNA, we start with U, T goes to A, C goes to G. The next 3 letters, A goes with U again, G with C, T with A. Next, C goes with G, T goes with A, T again goes with A. G goes with C, A to U, A to U. Lastly, T with A to U, A to U. So now we have our mRNA. I'm zooming in a little bit and get rid of this. Let's see, focus. There we go. So we have our strand of mRNA. So now we need to make a protein. There we go. So now with our protein, we're going to use a table. Remember, first letter's on the left, second letter's on the top, and then we go down to find the codon. So we have UAC. U starts here, all the way down. We get to A, and we find UAC, which is TYR, excuse me. The next one, UCA. U, find C, find A. That's SER, serine. Serine. Then we have GAA. G is here. A is this column. GAA, that's GUL, glutamate. And the next one, CUU. Again, C, we find U, CUU, which is LEU, leucine. And the last one is AUU. So the row for A, column for U, AUU is LILE, isoleucine. So now we have our strand, which is this. So if we go back to our slide, that is our answer. This is the protein that we make using this DNA strand. So make sure you practice this. Use that our virtual assignment we have for this week to help you practice this process. Now, almost done with it. With DNA, with gene expression. Gene expression, or when we read the DNA to make the protein, is highly regulated. Gene expression, the process of going from DNA to mRNA, mRNA to the actual protein, is very regulated because it requires a whole lot of energy. Cells want to save energy by only producing the needed proteins. So both prokaryotes and eukaryotes, regulate gene expression. They both want to regulate gene expression because they want to save energy and they want to save resources. Remember, when you make a protein, you're taking the three amino acids and putting them all together. You're taking the Lego pieces for a protein and put them together. You don't want to waste the amino acids. You don't want to waste energy. So gene expression is highly, highly regulated in the cell. The cell does not want to make extra proteins, extra material that it does not need. The last part of this unit is mutations. A mutation is a change in the cell's DNA sequence. So mutations come in several varieties. So whenever there's a change in the DNA sequence whenever there's a change in the DNA sequence, that's called a mutation. In this example here, this mutation caused the fly to grow legs where it should have antennas. So normally, this fruit fly, we have these small little appendages on its face that are the antennas. But the scientist was able to affect the gene for this antennas to make it legs instead. So that's an example of what a mutation can happen in a fly. Instead of it growing antennas on its face, it grows extra legs. And there's different types of mutations. So this table goes over all the different types. So when we talk about different types, we can talk about what's being changed and their effect. So, and the way I like to look at this table, and the way I like to think about mutations, is we start with the sentence. So I'm going to zoom in a little bit here. With the original sentence here, we have the one big fly had one red eye. That's the original sentence. I think the original sentence is the original gene. This is what the gene is supposed to say. We have different types of mutations here. A point mutation is a change in one or a few base pairs in the gene. So that can be like a substitution. So if we change just one letter in the sentence, that will be a point mutation. Where's my mouse? There it is. Let me get my laser pointer again. So a point mutation is a changing of one letter. So in this example, if we have an E, there's a mutation. We still get an E. It's still a mutation, but nothing happened. So say this. I'm going to go back real quick and zoom in. There we go. And we get a pen. I want to show you a different example of a point mutation. Hope it's not going to let me. Okay. So in a point mutation, say we have the one big fly had one red eye. Say in this mutation, instead of getting an E, I'm going to scratch this out. We get an A. There we go. Not the best drawing with the mouse. So if we change it to an A, it changes the meaning of this word. We have THA, one big fly had one red eye. So that would be a point mutation, changing that one letter to an A. So it can change the meaning of the sentence. It can change the meaning of the gene. So with a point mutation, if it's a substitution, which meaning it's only one letter is being changed, it can affect the whole protein. It may not, depending on how that amino acid is affected. So some mutations can cause disease. A single substitution in the gene for hemoglobin causes red blood cells to form abnormally, leading to sickle cell anemia. Let me go back to my pointer. So here's a gene we're looking at right now. This T is mutated to U, or mutated to A. So the DNA is changed from T to A. When it does that, instead of getting this GLU, we get VAL, VAL. So it's a point mutation because we're only changing one nucleotide in the DNA. And because that whole amino acid changes, what changes the whole structure of the cell. So this is what a normal red blood cell looks like. It's kind of this disc-like structure. But when we change this one base, instead of getting the disc, we get these weird structures instead. Because what happens in sickle cell anemia? It's a point mutation. We change one base pair in the DNA, and now we get a different shape for our red blood cells. So a mutation can cause disease. We also have what's called a frame shift mutation, which are large changes in the protein. So here, what we have is what's called an insertion. An insertion of one nucleotide changes the codon after the insertion. So remember, a codon is every three. So since the sentence the one big fly had one red eye, each word is 3 letters, so each one is a codon. But if we have an insertion, we are inserting a new letter, and we can change the whole meaning. So we can see here, if we insert one letter, take this Q, we get D1, and then everything after this is messed up. We are no longer getting the same sentence. It is a big change. We could have, if we insert 3 letters though, we get a shift in the protein. So instead of the one big fly had one red eye, we can get the one big wet fly had one red eye. So a new amino acid is inserted here, but if it's only one amino acid, it changes the whole protein. So frame shift affects multiple amino acids. So this is the original DNA here. So you can see if we, between the C and G, if we add a T, this whole reading frame changes. The reading frame is every 3 nucleotides. So instead of CAG, we get TGA. Instead of GAC next, we get CGA. So all these nucleotides are shifted and changed. If we had two, inserts 2 base pairs, two T's here, we still get a whole change in the frame. But if we insert 3, the reading is restored, because then we get GAC, GAC, GAC again, GAC, and so forth. And that's because, remember, a codon is 3 letters, 3 bases. So if we insert 3 bases, we're inserting a full amino acid here, and the rest of the amino acids are the same. Mutations can create genetic variety. We're going to see later on, I believe in this unit, that not all mutations are dangerous. Yes, mutations can cause disease. They can cause a disease, they can cause cancer, they can cause a lot of problems, but they're not always dangerous. Mutations can cause variations too. Mutations can cause different versions of alleles, which are alternate versions of the same gene. So we have different alleles, different genes for hair color, for eye color, for skin color, and so forth. So these are all different genes. They all function similarly, they all have the same function, but they have slightly different variation. The genetic variation is important for evolution, which we'll get into later in this class. In the example, plant breeders even introduce mutations to create new variations of plants. So there are some plants and some fruits that we eat that were heavily modified, heavily encouraged to mutate so we can get the plants, the food we eat. So wheat is an example of this, where wheat, it was bred in a way, it was made in a way where there's different mutations in the DNA. And this is through centuries and centuries of farmers doing this, where we get the type of wheat that we have now. So remember, mutations can be deadly. They can lead to disease, but they can also lead to new variations in an organism. They can be deadly, but they can also be helpful. All they are just a change in the DNA sequence. A change in the DNA sequence. And by changing the DNA sequence, you're changing the instructions for a protein. By changing the protein, you either can cause disease or can cause variation. Okay. So for this class, make sure you know for this week, make sure you know DNA, its structure, the basis. Make sure you know RNA, its structure, transcription, translation, and mutations.